Hello, and welcome to Calm Science Nights. I'm so glad you're here with me tonight. Before we begin our peaceful journey through the story of evolution, if you find comfort in drifting off to the soothing rhythm of scientific discovery, please consider subscribing to the channel. It helps us create more of these tranquil explorations together. Now, settle into your most comfortable position. Let the weight of the day lift from your shoulders. Tonight, we're going to travel through time, not with urgency or excitement, but with gentle curiosity, following the beautiful, slow unfolding of one of science's most profound ideas. We'll trace the story of evolutionary theory from its earliest whispers to its modern understanding, each fact a stepping stone in humanity's gradual awakening to the deep history of life on Earth. There's something deeply restful about this story, about how understanding grows slowly, how truth emerges gradually from observation and thought, how each generation of scientists built upon the insights of those who came before. Over the next 100 facts, we'll walk this peaceful path together, meeting the people who changed how we see ourselves and our place in nature, discovering the evidence that transformed our understanding of life itself. There's no need to remember everything tonight. In fact, there's no need to stay awake at all. Let these words wash over you like gentle waves, each fact a soft ripple in the stream of discovery, carrying you peacefully towards sleep. Just allow the story to unfold at its own unhurried pace, the way evolution itself unfolds, slowly, gradually, beautifully. Close your eyes now, and imagine yourself standing in a quiet English garden in the early 1800s, when the world still believed that every species had existed unchanged since the moment of creation, when the Earth itself was thought to be only a few thousand years old. Our story begins here, in this time of certainty, just before everything would begin to change. Long before Charles Darwin became a household name, before the word evolution entered common language, there were those who noticed things that didn't quite fit the story of an unchanging, recently created world. One of these observers was Charles Darwin's own grandfather, Erasmus Darwin, a physician and natural philosopher working in England in the late 1700s. Erasmus Darwin was a gentle, curious soul who studied everything from plants to medicine to the nature of life itself. In 1794, Erasmus Darwin published a book called Zoonomia, and in its pages, he suggested something quietly radical, that all warm-blooded animals might have arisen from one living filament, that life might have the power to transform itself over vast periods of time. He observed that animals undergo great changes during their development from embryo to adult, and he wondered whether species themselves might undergo similar transformations across the generations. He even noticed that organisms seemed to improve themselves through their own activities, a kind of self-improvement passed down through inheritance. These ideas were speculative, poetic, not yet supported by rigorous evidence, but they planted seeds. Erasmus Darwin died in 1802, when his grandson Charles was just 7 years old, never knowing that the child would carry these whispered questions into the realm of scientific revolution. The idea that life might change, might have a history, was beginning to stir in human consciousness like someone slowly waking from a long sleep. Around the same time Erasmus Darwin was writing, across the English Channel in France, another mind was contemplating similar mysteries. Jean-Baptiste Lamarck was a botanist who had turned his attention to the classification of invertebrate animals, those creatures without backbones that make up the vast majority of animal life. In his quiet, methodical study of shells and insects and worms, Lamarck noticed patterns that troubled him. In 1809, Lamarck published Philosophie zoologique, and in it, he proposed what may have been the first truly comprehensive theory of evolution. Lamarck observed that simpler organisms seemed to grade into more complex ones, that fossils showed a progression from simple ancient life to more complex modern life. He suggested that species were not fixed and unchanging, but rather transformed gradually over time, always moving toward greater complexity, greater perfection. Lamarck proposed a mechanism for this transformation, one that we now know is incorrect, but which was a sincere attempt to explain the mystery of change. He believed that animals could acquire new characteristics during their lifetime through use or disuse of their body parts, and that these acquired changes could be passed on to their offspring. The classic example, though Lamarck himself never used it, is the giraffe stretching its neck to reach high leaves, and this stretched neck somehow being inherited by its offspring, generation after generation, until long necks became characteristic of the species. This idea, the inheritance of acquired characteristics, would later be proven wrong. But Lamarck's deeper insight was correct and revolutionary. Species change over time, life has a history. The living world we see today is the result of a long process of transformation. For proposing this, Lamarck was largely ridiculed during his lifetime. He died in poverty and obscurity in 1829, blind and nearly forgotten. Yet his courage to question the fixity of species, to imagine that life itself had a story, was an essential step in the journey toward understanding evolution. While Lamarck struggled for recognition in France, in England a different kind of revolution was quietly unfolding, a revolution in understanding the Earth itself. A Scottish geologist named James Hutton had been studying rocks, river valleys, and mountains, and what he saw filled him with a sense of deep time that was shocking to his contemporaries. In the late 1700s, Hutton proposed what he called uniformitarianism, the idea that the same geological processes we observe today, like erosion, sedimentation, and volcanic activity, have been operating throughout Earth's history. This seems obvious now, but at the time, most people believed that Earth's features were created by sudden catastrophes, particularly the biblical flood. Hutton looked at the slow wearing away of rocks by rain and rivers, at the gradual buildup of sediment and layers, and he realized something profound. These processes are so slow that Earth must be vastly, almost inconceivably old. Hutton wrote that he could find no vestige of a beginning, no prospect of an end. The Earth, he suggested, operated in great cycles of destruction and renewal over periods of time so long that human history was but a brief flicker. When Hutton presented these ideas, they were met with resistance and skepticism. An Earth millions or billions of years old contradicted the accepted biblical chronology that placed creation at around 4004 BC. But Hutton's ideas would be carried forward by another geologist, Charles Lyell, who was born the year Hutton died. Lyell spent decades studying geological formations across Europe, and between 1830 and 1833, he published his masterwork, Principles of Geology. In clear, compelling prose, Lyell laid out the evidence for an ancient Earth shaped by gradual processes operating over immense spans of time, the same processes we can observe today, working slowly, patiently, century after century, millennium after millennium. Lyell's work was revolutionary not just for geology but for biology, because it provided something essential that evolution would need: time. If species were to transform gradually, naturally, they would need vast ages in which to do so. Lyell's ancient Earth, with its millions of years of history written in rock layers, provided exactly the temporal canvas on which evolution could paint its slow masterpiece. A young man named Charles Darwin carried the first volume of Lyell's Principles of Geology with him when he boarded a ship called HMS Beagle in December 1831. Darwin was just 22 years old, fresh out of Cambridge University, where he had studied to become a clergyman. He had no intention of revolutionizing biology. He simply loved nature, loved collecting beetles and observing birds, and when he was offered the position of gentleman's companion and naturalist aboard the Beagle's 5-year surveying voyage around the world, he accepted, imagining it as an adventure, a chance to see exotic lands and collect interesting specimens. The voyage of the Beagle would last from 1831 to 1836, and during those 5 years, Darwin would witness things that would slowly, gradually transform his understanding of life. The journey wasn't comfortable. Darwin suffered terribly from seasickness, but every time the ship made landfall, he would venture out to explore, to observe, to collect, to wonder. In South America, he found fossils of giant extinct animals that resembled, but were much larger than, the living animals of the region. Why would extinct species resemble the living species of the same area? This seemed odd if all species were created separately and independently. He observed how species seemed to change gradually as he traveled north to south along the continent. He noticed that different regions, even when they had similar climates, were inhabited by different but related species. And then, in September 1835, the Beagle reached a small cluster of volcanic islands about 600 miles off the coast of Ecuador, the Galapagos Islands. These islands, so remote and so strange, would become central to Darwin's thinking, though he didn't realize it at the time. On the Galapagos, Darwin encountered giant tortoises, marine iguanas, and most famously, finches, small birds that varied from island to island in the shapes of their beaks. At first, Darwin didn't pay special attention to these variations. He didn't even carefully label which island each bird came from. But later, as he reflected on his observations, the pattern began to trouble him. Why would each island have its own distinct forms? Why would these forms be similar to each other and to mainland species, yet distinct? If all species were created independently, why this pattern of variation? Why not completely different creatures on each island? And so our story has begun in the soft dawn of evolutionary thought, with Erasmus Darwin in the 1790s, quietly suggesting in his book Zoonomia that all warm-blooded animals might arise from one living filament, that life might transform itself over vast time, observing that organisms change greatly from embryo to adult and wondering whether species might change across generations through a kind of self-improvement passed down through inheritance. These were seeds planted, speculative and poetic, not yet proven, but beginning to stir. We met Jean-Baptiste Lamarck, who in 1809 published Philosophie zoologique, proposing perhaps the first comprehensive evolutionary theory, observing that simpler organisms grade into complex ones, that fossils show progression from simple to complex, suggesting species transform gradually toward greater complexity. His mechanism, inheritance of acquired characteristics, animals developing traits through use or disuse passed to offspring, would prove incorrect, but his deeper truth was revolutionary. species change over time, life has a history. Lamarck died in 1829 in poverty, blind and forgotten, ridiculed for questioning the fixity of species, yet his courage was essential. We discovered James Hutton's geological revolution in the late 1700s, uniformitarianism, the idea that current geological processes operated throughout Earth's history, that slow erosion, sedimentation, and volcanic activity require vast time. His observation that he could find no vestige of a beginning, no prospect of an end suggested an Earth millions or billions of years old, contradicting the accepted 4004 BC creation date. This ancient Earth operating in great cycles of destruction and renewal was necessary for evolution. Species transforming gradually would need vast ages. We learn Charles Lyell carried Hutton's work forward, publishing Principles of Geology between 1830 and 1833, laying out evidence for an ancient Earth shaped by gradual processes over immense spans, the same processes observable today working slowly across millennia. Lyell's ancient Earth with millions of years written in rock layers provided the temporal canvas evolution needed, and young Charles Darwin carried Lyell's first volume aboard HMS Beagle in December 1831. We followed Darwin boarding the Beagle at 22, fresh from Cambridge where he studied to become a clergyman, accepting the position of gentleman's companion and naturalist for a 5-year surveying voyage. The 1831 to 1836 journey transformed his understanding. In South America he found giant extinct animal fossils resembling but larger than living regional animals, observed species changing gradually traveling north to south, noticed different regions with similar climates had different but related species. We arrived at September 1835, when the Beagle reached the Galapagos Islands, 600 miles off Ecuador's coast, remote volcanic islands where Darwin encountered giant tortoises, marine iguanas, and finches varying island to island in beak shapes. Initially he didn't pay special attention or carefully label which island each came from, but later the pattern troubled him. Why distinct forms on each island? Why similar to each other in mainland species yet distinct? If species independently created, why this variation pattern instead of completely different creatures? Each fact a gentle step in understanding slow emergence, each observation a quiet question beginning to form. The world waking gradually to the deep history of life, to the possibility of change, to the vastness of time. Questions stirring like the first light of dawn, soft and patient, in no hurry to reach conclusions. Feel yourself in that gentle time of awakening, when old certainties begin to soften, when new possibilities whisper at the edges of consciousness. There's nothing to solve tonight, nothing to grasp. Just rest in the slow unfolding of discovery, in the peaceful rhythm of minds opening to truth, generation after generation, slowly, beautifully, like flowers opening to the sun. As we continue our peaceful journey through the story of evolution, we find Charles Darwin returning home to England in October 1836, after 5 years aboard the Beagle. He was no longer the young man who had set sail. His mind was full of specimens, observations, questions. But he didn't immediately announce a revolutionary theory. Instead, he did something that would characterize his entire approach to science. He began to think, carefully and methodically, about what he had seen. Darwin settled into a quiet life in London, organizing his collections, writing up his voyage observations, and beginning the slow, private work of puzzling through the mystery of species. He filled notebook after notebook with his thoughts, drawing diagrams, questioning, considering possibilities. In July 1837, less than a year after returning home, Darwin opened a new notebook and wrote at the top, Zoonomia, the same title his grandfather had used. He was consciously taking up the question of life's mutability, but he would approach it with a rigor and evidence his grandfather had never attempted. In these private notebooks, we can watch Darwin's thinking evolve. He sketched a branching diagram, perhaps the first evolutionary tree, showing how species might split and diverge from common ancestors over time. This was a profound insight. Life's history wasn't a ladder of progress from simple to complex, as Lamarck had imagined, but a branching tree, with each twig representing a species, each branch point representing a common ancestor. Some branches flourish and split further, others end in extinction. This tree of life, drawn in a small notebook that no one else would see for decades, would become one of the most powerful images in all of biology. But Darwin still didn't have a mechanism. He didn't understand how species changed, what force drove them to adapt to their environments. And then, in September 1838, almost by accident, he found the key. Darwin was reading widely, as was his habit, and he picked up a book by Thomas Malthus called An Essay on the Principle of Population. Malthus wasn't writing about biology at all, he was writing about economics and human society. Malthus observed that human populations tend to grow exponentially, doubling every generation, while food supplies grow only arithmetically, at a much slower rate. The implication, Malthus argued grimly, was that populations would always outstrip resources, leading to competition, famine, war, disease, a constant struggle for survival. Most people reading Malthus found his vision depressing, even horrifying. But Darwin saw something different. He realized that this principle applied not just to humans but to all living things. Every species produces more offspring than can possibly survive. A single oyster can produce millions of eggs, an oak tree thousands of acorns. Yet population sizes remain relatively stable. This means that most offspring die. There is a struggle for existence. And here was the crucial insight. In this struggle, any advantage, however slight, might make the difference between survival and death. If individuals vary, and Darwin knew from his observations that they do, and if some of those variations are heritable, and Darwin knew from animal breeding that they are, then individuals with advantageous variations would be more likely to survive and reproduce. Over many generations, these small advantages would accumulate, and the population would gradually change, adapting to its environment. Darwin called this process natural selection, nature selecting which individuals survive and reproduce, the same way a farmer selects which animals to breed, but working slowly, over countless generations. It wasn't a conscious process, wasn't directed toward any goal. It was simply the inevitable result of variation, inheritance, and differential survival. The beautiful adaptations of nature, the orchid's flower, the bird's wing, the eye's complexity, weren't designed but rather emerged gradually through this patient, unconscious process. Darwin had his theory by 1838, just 2 years after returning from the Beagle voyage. He was 30 years old. And then he did something surprising. He didn't publish it. For 20 years, Darwin kept his theory largely private, sharing it only with a few trusted colleagues. Why this long delay? partly, Darwin wanted to be absolutely certain, to gather overwhelming evidence before making such a revolutionary claim. He knew the idea would be controversial, even shocking. The notion that humans and animals shared common ancestors, that we were part of nature rather than separate from it, contradicted deeply held religious beliefs. But there was another reason for Darwin's caution, one that reveals much about the man. Darwin's wife Emma was deeply religious, and he knew his theory would trouble her, might suggest to her that life had no divine purpose. Darwin loved his wife dearly, and the thought of causing her pain made him reluctant to publish. So instead of rushing into print, Darwin spent those 20 years doing meticulous research, studying everything from barnacles to orchids, from earthworms to domestic pigeons, always gathering more evidence, always strengthening his argument. During these quiet years, Darwin moved his family to a house called Down House in the Kent countryside, about 16 miles from London. There, in peaceful isolation, surrounded by gardens and fields, Darwin lived the life of a country gentleman naturalist. He was often ill, suffering from mysterious symptoms that plagued him for most of his adult life, but he worked steadily, methodically, in his study overlooking the garden. He dissected barnacles for 8 years, describing every species he could find, learning about variation and adaptation in painstaking detail. To his children, he seemed like any other father, walking in the garden, playing games, but he was quietly assembling one of the most important scientific arguments in history. Meanwhile, on the other side of the world, another naturalist was having similar thoughts. Alfred Russel Wallace was a young man from a modest background who made his living collecting specimens in exotic locations and selling them to museums and collectors. In the 1850s, Wallace was working in the Malay Archipelago, the islands of Southeast Asia, enduring hardship, disease, and danger to collect birds, insects, and other specimens in some of the most remote jungles on Earth. Wallace was also a deep thinker, and like Darwin, he was puzzled by the distribution and variation of species. In February 1858, while recovering from a fever on an island called Ternate, Wallace had a sudden flash of insight. He too had read Malthus, and he too saw that the struggle for existence could drive species change. In a burst of inspiration during his illness, Wallace sketched out the theory of evolution by natural selection, the same theory Darwin had been carefully developing for 20 years. Wallace didn't know that Darwin had already arrived at the same conclusions. He wrote up his ideas in a short essay and, with touching trust, sent it to Darwin asking for his opinion and help in getting it published. When Darwin received Wallace's letter in June 1858, he was stunned. Here was his own theory, clearly articulated, about to be published by someone else. After 20 years of cautious work, Darwin faced the prospect of losing priority for his greatest discovery. Darwin's friends, the geologist Charles Lyell and the botanist Joseph Hooker, helped arrange a compromise. On July 1, 1858, both Darwin's and Wallace's papers were read at a meeting of the Linnean Society in London. Neither Darwin nor Wallace was present. Darwin's youngest son had just died of scarlet fever, and Wallace was still in the distant islands, unaware of the stir his letter had caused. The presentation was received politely but without great excitement. The president of the Linnean Society later remarked that the year had not been marked by any revolutionary discoveries. But the papers had been published, priority was established, and Darwin now felt compelled to finally produce the full exposition of his theory. He called it his abstract, though it would turn out to be a substantial book, and he worked on it intensely for the next 13 months. On November 24, 1859, On the Origin of Species by Means of Natural Selection was published. The first printing of 1,250 copies sold out on the first day. The book was revolutionary, yet Darwin wrote it in a careful, gentle style, piling evidence upon evidence, anticipating objections, addressing difficulties honestly. He avoided discussing human evolution directly, except for one carefully neutral sentence near the end: light will be thrown on the origin of man and his history. But everyone understood the implication. If all species shared common ancestors, if life had evolved gradually from simple forms to complex ones, then humans too were part of this grand story, descendants of earlier animals, cousins to the apes. And so we followed Darwin home to England in October 1836 after 5 years aboard the Beagle, no longer the young man who sailed, mind full of specimens and questions. He settled in London organizing collections and writing, beginning slow private work puzzling through species mystery. July 1837, less than a year home, Darwin opened a new notebook titled Zoonomia, his grandfather's title, consciously taking up life's mutability question but with unprecedented rigor. We watched his thinking evolve in private notebooks, sketching a branching diagram, perhaps the first evolutionary tree, showing how species might split and diverge from common ancestors. Profound insight. Life's history wasn't a ladder from simple to complex as Lamarck imagined, but a branching tree, each twig a species, each branch point a common ancestor, some branches flourishing and splitting, others ending in extinction. This tree of life, drawn unseen for decades, would become biology's most powerful image. We discovered September 1838, Darwin reading Thomas Malthus's essay on the principle of population, economics and human society, not biology. Malthus observed human populations grow exponentially, doubling each generation, while food grows arithmetically, slower rate, implying populations outstrip resources leading to competition, famine, war, disease, constant survival struggle. Darwin saw this applied to all life. Every species produces more offspring than can survive: oyster, millions of eggs; oak, thousands of acorns, yet populations remain stable, meaning most offspring die, struggle for existence. We learned Darwin's crucial insight: in this struggle, any slight advantage makes survival-death difference. Individuals vary. Darwin knew from observations, some variations heritable, knew from animal breeding, so individuals with advantageous variations more likely to survive and reproduce. Over generations, small advantages accumulate, populations gradually change, adapting to environment, natural selection, nature selecting survivors like a farmer selects breeding animals, but working slowly over countless generations, unconscious, inevitable result of variation, inheritance, differential survival. We found Darwin had his theory by 1838, age 30, 2 years after returning, then surprisingly didn't publish for 20 years. Why? Wanted absolute certainty, overwhelming evidence before revolutionary claim, new idea would be controversial, shocking, humans and animals sharing common ancestors, humans part of nature not separate, contradicting religious beliefs. Also Darwin's wife Emma deeply religious, theory would trouble her, suggest life has no divine purpose, loving her dearly, reluctant to cause pain. We followed Darwin's quiet 20 years, moved family to Down House in Kent countryside, 16 miles from London, peaceful isolation surrounded by gardens and fields, country gentleman naturalist life, often ill with mysterious symptoms but worked steadily, methodically, dissected barnacles for 8 years describing every species, learning about variation and adaptation in painstaking detail. To children seemed like any father walking gardens and playing games, but quietly assembling one of history's most important scientific arguments. We met Alfred Russel Wallace, young man from modest background making living collecting specimens in exotic locations, selling to museums and collectors, 1850s working in Malay Archipelago, Southeast Asia Islands, enduring hardship, disease, danger collecting birds and insects in remote jungles, also puzzled by species distribution and variation. We discovered February 1858, Wallace recovering from fever on Ternate Island had sudden insight, also read Malthus, saw survival struggle could drive species change, burst of inspiration during illness, sketched evolution by natural selection theory, same theory Darwin developed 20 years, didn't know Darwin's work, wrote essay, sent to Darwin asking opinion and publication help. Darwin received June 1858, stunned, his theory about to be published by someone else after 20 years cautious work. We learned Darwin's friends Lyell and Hooker arranged compromise. July 1, 1858, both Darwin's and Wallace's papers read at Linnean Society London meeting, neither present. Darwin's youngest son just died of scarlet fever, Wallace still in distant islands unaware, received politely without great excitement. President later remarked you had no revolutionary discoveries, but papers published, priority established. Darwin compelled to produce full exposition. We arrived at Darwin's intense 13-month work on his abstract, substantial book, November 24, 1859, On the Origin of Species by Means of Natural Selection published, first printing 1,250 copies sold out first day, revolutionary yet careful, gentle style, evidence upon evidence, anticipating objections, addressing difficulties honestly, avoided direct human evolution discussion except one sentence near end, light will be thrown on the origin of man and his history, but everyone understood implication, humans part of grand story, descendants of earlier animals, cousins to apes. Each fact a quiet step forward in understanding's emergence, each piece of the puzzle fitting gently into place. Darwin thinking, questioning, gathering evidence with patience, with care, with no hurry to reach conclusions. 20 years of quiet work, of letting truth reveal itself slowly, like dawn spreading gradually across the landscape. Rest in this patient unfolding, this gentle rhythm of discovery. No rush. No urgency, just the slow, beautiful emergence of understanding. Let these facts carry you softly, like leaves floating on a calm stream, drifting peacefully towards sleep. As we continue our gentle journey through evolution's story, we arrive at the moment when Darwin's carefully constructed theory met the world. The publication of On the Origin of Species in November 1859 created ripples that would spread outward in all directions, touching science, religion, philosophy, and society itself. But the response was not what we might expect, not universal outrage, not immediate acceptance, but something more complex, more nuanced, more human. The scientific community's reaction was mixed and thoughtful. Many scientists were immediately convinced or at least intrigued. Thomas Henry Huxley, a brilliant anatomist and fierce debater, reportedly said upon reading the book, "How extremely stupid not to have thought of that." Huxley would become Darwin's most vocal public defender, willing to engage in debates that the shy, retiring Darwin himself avoided. Huxley was so vigorous in defending evolution that he became known as Darwin's bulldog, though Darwin himself never asked for such aggressive advocacy. But not all scientists were convinced. Richard Owen, Britain's leading anatomist and the man who had coined the word dinosaur, became one of Darwin's most persistent critics. Owen was a complex figure, brilliant, ambitious, prone to claiming others' discoveries as his own. He accepted that species had changed over time, but he could not accept Darwin's mechanism of natural selection. Owen believed there must be some divine plan, some directing intelligence behind the patterns of life. The debate between Owen and Huxley would become personal and bitter, a reminder that scientific disagreements are conducted by human beings with egos, ambitions, and deeply held beliefs. The famous confrontation came in June 1860, just 7 months after The Origin was published, at a meeting of the British Association for the Advancement of Science in Oxford. Bishop Samuel Wilberforce, who had been coached by Richard Owen, gave a long speech criticizing Darwin's theory. The bishop was known for his eloquence and his sarcastic wit. He was called Soapy Sam because of his smooth speaking style. At the end of his speech, Wilberforce turned to Huxley and asked, mockingly, whether it was through his grandfather or his grandmother that he claimed descent from an ape. Huxley reportedly whispered to a colleague, "The Lord hath delivered him into mine hands," and then rose to respond. In careful, measured tones, Huxley said he would not be ashamed to have an ape for an ancestor, but he would be ashamed to be connected with a man who used his gifts to obscure the truth. The audience gasped. Some cheered. One woman reportedly fainted at the impiety of it all. The Oxford debate became legendary, a symbolic moment when science confronted religious authority, though the reality was probably less dramatic than the legend suggests. Most accounts were written years later, memories colored by what people wanted to believe had happened. What's often forgotten is that the debate wasn't really about whether evolution had occurred. By 1860, many scientists and even some clergy accepted that species had changed over time. The real controversy was about mechanism and meaning. Could natural selection, an unconscious, undirected process, really account for the exquisite complexity of life? And more troubling to many, if humans evolved through natural selection, did that mean we were merely animals, that our existence had no special purpose, no divine intention? Darwin himself remained largely above the fray, continuing his quiet work at Down House. He suffered from anxiety and illness, possibly exacerbated by the stress of public controversy, and he left the debating to others like Huxley. But he continued to gather evidence, to refine his arguments, to address objections. One persistent problem troubled him and his critics alike, the question of heredity. Darwin's theory required that variations be passed from parent to offspring, but no one understood how inheritance actually worked. Darwin himself proposed a theory called pangenesis, suggesting that every part of the body produced tiny particles called gemmules that carried hereditary information. This theory was wrong, and it would eventually be forgotten, but it showed Darwin grappling honestly with a genuine gap in his understanding. The irony is that the answer to the heredity question was being discovered at exactly the same time, in a quiet monastery garden in what is now the Czech Republic. A monk named Gregor Mendel was conducting meticulous experiments with pea plants, carefully crossbreeding them and counting the traits of the offspring. Between 1856 and 1863, through thousands of crosses and careful statistical analysis, Mendel discovered the fundamental laws of inheritance, that traits are passed down in discrete units, that some traits are dominant while others are recessive, that heredity follows mathematical patterns. Mendel published his results in 1866 in a regional scientific journal. He sent copies to prominent botanists, including one who corresponded with Darwin. But Mendel's work was ignored, overlooked, forgotten. Perhaps it was too mathematical for biologists of the time, too abstract, buried in an obscure journal. Perhaps the scientific world simply wasn't ready to recognize its importance. Mendel died in 1884, never knowing that his careful experiments had laid the foundation for the science of genetics. His work would remain hidden for over 30 years, waiting to be rediscovered at the dawn of the 20th century. Meanwhile, Darwin turned his attention to a question he had carefully avoided in The Origin of Species: what about humans? In 1871, 12 years after The Origin, Darwin published The Descent of Man, and Selection in Relation to Sex. Here, he laid out the evidence that humans had evolved from earlier primates, that we shared common ancestors with apes, that our physical and mental characteristics could be explained by natural selection and sexual selection, the process by which traits evolve because they help in attracting mates or competing for them. The book was comprehensive and careful, filled with comparative anatomy, observations of behavior, and thoughtful analysis. Darwin argued that the difference between human and animal minds was one of degree, not kind, that animals showed rudimentary forms of reasoning, emotion, even morality, and that human faculties had evolved gradually from these simpler foundations. He discussed the evolution of moral sense, of language, of our sense of beauty. And he introduced the concept of sexual selection to explain traits that seemed to have no survival value: the peacock's tail, the lion's mane, human beardlessness and hairlessness. The response to The Descent of Man was calmer than many expected. Perhaps the public had had 12 years to get used to the idea of evolution. Perhaps Darwin's careful, respectful tone made the argument more acceptable. There was criticism, of course, particularly from religious quarters, but there was also acceptance and even enthusiasm. The idea that humans were part of nature, subject to the same laws as other organisms, was gradually becoming part of the scientific worldview, even if it made some people uncomfortable. But accepting evolution and understanding evolution were two different things. Throughout the late 1800s, debates raged about the mechanisms, the speed, the patterns of evolutionary change. Some scientists emphasized gradual change, others sudden jumps. Some focused on natural selection, others on different proposed mechanisms. The geologist Charles Lyell, Darwin's old friend and mentor, accepted evolution but struggled to accept that humans were truly just another animal. Even those sympathetic to Darwin's ideas often made an exception for human beings, wanting to preserve something special, something transcendent about our species. By the time Darwin died in 1882, at the age of 73, evolution was widely accepted in the scientific community, but the full synthesis of his ideas was still decades away. Darwin was buried in Westminster Abbey, near the grave of Isaac Newton, a recognition of his monumental contribution to human knowledge. The shy naturalist who had spent 20 years hesitating to publish his theory was laid to rest as one of the greatest scientists who had ever lived. And so we witnessed the world's response to The Origin, published November 1859, creating complex ripples through science, religion, philosophy, society. Thomas Henry Huxley, brilliant anatomist, upon reading said how extremely stupid not to have thought of that, became Darwin's most vocal defender, Darwin's bulldog, engaging in debates the shy Darwin avoided. But Richard Owen, Britain's leading anatomist who coined dinosaur, became persistent critic, accepted species change but not natural selection mechanism, believed must be divine plan, directing intelligence. Debate between Owen and Huxley became personal and bitter. We arrived at the famous June 1860 confrontation, 7 months after Origin publication, at British Association meeting in Oxford. Bishop Samuel Wilberforce, Soapy Sam for smooth eloquence, coached by Owen, gave long critical speech, then mockingly asked Huxley whether through grandfather or grandmother he claimed ape descent. Huxley whispered, "The Lord hath delivered him into mine hands," responded he wouldn't be ashamed of ape ancestor but would be ashamed of man using gifts to obscure truth. Audience gasped, some cheered, one woman reportedly fainted. Oxford debate became legendary symbolic moment, though reality probably less dramatic than legend. We learned the real 1860 controversy wasn't whether evolution occurred. Many scientists and some clergy accepted species change by then, but about mechanism and meaning. Could unconscious, undirected natural selection account for life's exquisite complexity? If humans evolved through natural selection, were we merely animals, our existence without special purpose or divine intention? We found Darwin remained above the fray at Down House, suffering anxiety and illness possibly from controversy stress, leaving debating to others like Huxley, continued gathering evidence, refining arguments, addressing objections. Persistent problem, heredity question. Theory required variations passed parent to offspring, but no one understood how inheritance worked. Darwin proposed pangenesis. Every body part produces tiny gemmules carrying hereditary information. Theory was wrong, eventually forgotten, but showed honest grappling with genuine understanding gap. We discovered the irony. Heredity answer being discovered simultaneously in quiet monastery garden, now Czech Republic. Monk Gregor Mendel conducting meticulous peat plant experiments, carefully crossbreeding and counting offspring traits, 1856 to 1863 through thousands of crosses and statistical analysis, discovered fundamental inheritance laws, traits passed in discrete units, some dominant others recessive, heredity follows mathematical patterns. We learned Mendel published results 1866 in Regional Scientific Journal, sent copies to prominent botanists including one corresponding with Darwin, but work was ignored, overlooked, forgotten, perhaps too mathematical for biologists, too abstract, obscure journal, or scientific world not ready. Mendel died 1884 never knowing his experiments laid genetics foundation. Work remained hidden over 30 years, waiting rediscovery at 20th century dawn. We followed Darwin turning to carefully avoided question, humans. 1871, 12 years after Origin, published The Descent of Man, and Selection in Relation to Sex, evidence humans evolved from earlier primates, share common ancestors with apes, physical and mental characteristics explained by natural selection and sexual selection, traits evolving to attract mates or compete for them, comprehensive, careful, filled with comparative anatomy, behavioral observations, thoughtful analysis. We found Darwin argued human-animal mind difference was degree not kind. Animals show rudimentary reasoning, emotion, even morality. Human faculties evolved gradually from simpler foundations. Discussed moral sense evolution, language, beauty sense, introduced sexual selection for traits without survival value, peacock tail, lion mane, human beardlessness/hairlessness, response calmer than expected. Public had 12 years adjusting to evolution. Darwin's careful, respectful tone made argument more acceptable, criticism from religious quarters but also acceptance and enthusiasm. We learned accepting evolution differed from understanding evolution. Late 1800s debates raged about mechanisms, speed, change patterns. Some emphasized gradual change, others sudden jumps. Some focused on natural selection, others on different proposed mechanisms. Even sympathetic figures like Lyell accepted evolution but struggled accepting humans as just another animal, wanting to preserve something special, transcendent about our species. We arrived at Darwin's death 1882, age 73, evolution widely accepted in scientific community but full synthesis still decades away. Buried in Westminster Abbey near Isaac Newton, recognition of monumental contribution to human knowledge. Shy naturalist who hesitated 20 years to publish laid to rest as one of greatest scientists who ever lived. Each fact a gentle reminder of how understanding unfolds through human lives, human debates, human struggles. Ideas meeting resistance and acceptance, truth emerging gradually through conversation and conflict, brilliant minds building on each other's work, sometimes without knowing it. Mendel's peas growing in a quiet garden while Darwin puzzles over heredity. Huxley defending ideas in public while Darwin works in private. Knowledge advancing through this beautiful, messy, profoundly human process. Rest in this understanding now, this recognition that discovery is patient, gradual, full of stops and starts. Let yourself drift like Mendel's pea pollen on a gentle breeze, like Darwin's thoughts wandering through his garden, peaceful and unhurried, moving slowly towards sleep. As we continue our peaceful journey through evolution's story, we move into a new era, the turn of the 20th century, when Darwin's theory would be tested, challenged, and ultimately strengthened by discoveries he could never have imagined. The decades following Darwin's death in 1882 were a time of confusion and debate, often called the eclipse of Darwinism, when natural selection fell somewhat out of favor and scientists proposed alternative mechanisms for evolution. During these years, many biologists accepted that evolution had occurred but doubted whether natural selection was powerful enough to be the primary driving force. Some favored Lamarck's old idea of inheritance of acquired characteristics. Others proposed that evolution proceeded in great leaps rather than gradual changes, or that organisms had an inherent tendency to evolve in certain directions, a kind of internal drive toward complexity. These were the dark ages for Darwin's theory, not because evolution was rejected, but because the elegant simplicity of natural selection was temporarily obscured by a proliferation of alternative ideas. Then, in 1900, something remarkable happened, something that would eventually rescue Darwin's theory and transform it into modern evolutionary biology. Three botanists working independently in 3 different countries, Hugo de Vries in the Netherlands, Karl Correns in Germany, and Erich von Schermach in Austria, all rediscovered Mendel's long-forgotten papers on inheritance. Each had been conducting breeding experiments and arrived at similar conclusions to Mendel, and each, searching the scientific literature, found that a monk in Moravia had anticipated their findings by 35 years. The rediscovery of Mendel's work was like finding a missing puzzle piece. Suddenly, heredity had laws, mathematical patterns, predictability. Traits were passed down in discrete units, which would soon be called genes, and these units maintained their integrity generation after generation. This was exactly what Darwin's theory needed, a mechanism for heredity that preserved variations rather than blending them away. Yet ironically, the early geneticists saw themselves as opponents of Darwinism, not supporters. The reason for this opposition was mutations. Hugo de Vries, studying evening primrose plants, observed what he thought were sudden, dramatic changes producing new varieties in a single generation. He called these changes mutations and proposed that evolution occurred through these large jumps rather than through Darwin's gradual accumulation of small variations. De Vries believed he had found a better mechanism than natural selection, evolution by mutation, creating new species in sudden leaps. For a time, this mutationist view was highly influential, and it seemed to many scientists that genetics had disproven Darwinism. But this conflict was based on a misunderstanding that would take decades to resolve. The early geneticists were studying heredity in the laboratory, looking at controlled crosses and discrete traits. The naturalists and paleontologists were studying variation in wild populations and patterns in the fossil record. These two groups were looking at different scales, different timescales, using different methods, and they struggled to communicate with each other. What was needed was someone who could bridge these two worlds, who understood both genetics and natural selection, who could show how they fit together rather than contradicting each other. That synthesis began to emerge in the 1920s and 1930s, through the work of mathematical biologists who created what came to be called population genetics. Three men were central to this revolution: Ronald Fisher in England, J.B.S. Haldane also in England, and Sewell Wright in the United States. These scientists were comfortable with mathematics in a way most biologists weren't, and they used mathematical models to understand how genes behave in populations over time. Fisher, Haldane, and Wright showed, through elegant mathematical reasoning, that Mendelian genetics and natural selection were not only compatible but complementary. Small mutations affecting survival or reproduction, even very slightly, could gradually spread through populations over generations through natural selection. Evolution didn't require large dramatic jumps. Small genetic changes, accumulated over time, were sufficient. The mathematics proved that natural selection was indeed a powerful force capable of driving evolutionary change, just as Darwin had proposed, and that Mendelian inheritance provided exactly the mechanism Darwin had lacked. In 1930, Fisher published The Genetical Theory of Natural Selection, a dense mathematical treatise that laid out the theoretical foundation for understanding evolution as a change in gene frequencies in populations over time. Wright developed the concept of adaptive landscapes, imagining evolution as populations climbing fitness peaks in a multidimensional landscape of possible genetic combinations. Haldane calculated how quickly advantageous genes would spread and showed that even small selective advantages could have large effects over evolutionary time. This work was abstract, mathematical, often difficult even for professional biologists to follow, but it was rigorous and powerful. The next step was to connect this mathematical theory to biological reality, to show that the principles worked not just on paper but in living organisms. This is where the architects of the modern synthesis came in, biologists who integrated genetics, natural selection, paleontology, systematics, and botany into a unified framework. The Russian-American geneticist Theodosius Dobzhansky was one of the most important of these synthesizers. Dobzhansky had trained in Russia studying ladybird beetles, then moved to the United States in 1927 to work with Thomas Hunt Morgan, the pioneering fruit fly geneticist. Dobzhansky combined Morgan's genetic techniques with Fisher's theoretical insights and his own deep understanding of natural populations. In 1937, he published Genetics and the Origin of Species, a book that translated the mathematical theory of population genetics into language biologists could understand, filled with concrete examples from his own research on fruit fly populations in the wild. Dobzhansky showed that natural populations harbor enormous genetic variation, far more than anyone had suspected. This variation was the raw material for evolution. He demonstrated that even seemingly trivial genetic differences could affect survival and reproduction in measurable ways. He studied how populations adapt to local conditions, how geographic barriers lead to genetic divergence, how new species form through the accumulation of genetic incompatibilities. Dobzhansky made evolution tangible and testable, moving it from abstract theory to observable process. Around the same time, other biologists were making similar syntheses in their own fields. Ernst Mayr, a German ornithologist who had studied bird species in New Guinea and the Solomon Islands, published Systematics and the Origin of Species in 1942. Mayr brought a naturalist's eye to the question of how species form. He emphasized that species aren't just groups of similar organisms but populations that are reproductively isolated. They don't interbreed with other species. This biological species concept helped clarify what a species actually is and how new species arise. Mayr argued that new species typically form when populations become geographically separated, perhaps by a mountain range rising, a river changing course, or a few individuals colonizing an island. In isolation, these separated populations evolve independently, accumulating different mutations, adapting to different local conditions. Eventually, they become so different that even if they come back into contact, they can no longer successfully interbreed. They have become separate species. This process, called allopatric speciation, became the dominant model for how biodiversity arises. The paleontologist George Gaylord Simpson added the perspective of deep time to the synthesis. In 1944, he published Tempo and Mode in Evolution, examining what the fossil record revealed about evolutionary patterns. Simpson showed that the fossils confirmed the general picture emerging from genetics and field biology. Evolution occurred at varying rates, sometimes slowly and gradually, sometimes more rapidly during periods of environmental change or when organisms colonized new habitats. The gaps in the fossil record that had troubled Darwin could be explained by the rarity of fossilization and the incompleteness of our sampling, not by mysterious jumps or divine intervention. And so we entered the 20th century's opening decades, the eclipse of Darwinism, when many biologists accepted evolution but doubted natural selection's power as primary driving force. Some favored Lamarck's acquired characteristics inheritance, others proposed great leaps rather than gradual changes, or inherent tendency toward complexity, internal drive. These Dark Ages for Darwin's theory saw natural selection's elegant simplicity obscured by proliferating alternative ideas. We discovered 1900's remarkable event, 3 botanists working independently in 3 countries, Hugo de Vries, Netherlands, Karl Correns, Germany, Erich von Schermach, Austria, all rediscovered Mendel's long-forgotten inheritance papers. Each conducting breeding experiments arrived at similar conclusions. Each searching literature found a Moravian monk had anticipated their findings by 35 years. We learned Mendel's rediscovery was like finding missing puzzle piece. Suddenly heredity had laws, mathematical patterns, predictability, traits passed in discrete units, soon called genes, maintaining integrity generation after generation, exactly what Darwin's theory needed, mechanism preserving variations rather than blending away. Yet ironically, early geneticists saw themselves as Darwinism opponents, not supporters. We found opposition reason was mutations. De Vries, studying evening primrose, observed sudden dramatic changes producing new varieties in single generation, called these mutations. Proposed evolution occurred through large jumps rather than Darwin's gradual small variation accumulation. Mutationist view highly influential, seemed genetics had disproven Darwinism, but conflict based on misunderstanding taking decades to resolve. Early geneticists studied laboratory heredity and discrete traits. Naturalists and paleontologists studied wild population variation and fossil patterns. Different scales, timescales, methods struggled to communicate. We met the bridge builders, 1920s to 1930s mathematical biologists creating population genetics, Ronald Fisher, England, J.B.S. Haldane, England, Sewell Wright, United States, comfortable with mathematics unlike most biologists, used mathematical models understanding how genes behave in populations over time. We learned Fisher, Haldane, Wright showed through elegant mathematical reasoning that Mendelian genetics and natural selection were compatible and complementary. Small mutations affecting survival/reproduction even slightly could gradually spread through populations over generations through natural selection. Evolution didn't require large jumps. Small genetic changes accumulated over time were sufficient. Mathematics proved natural selection powerful force capable of driving change as Darwin proposed. Mendelian inheritance provided mechanism Darwin lacked. We discovered 1930 Fisher published The Genetical Theory of Natural Selection, dense mathematical treatise laying theoretical foundation for understanding evolution as gene frequency changes in populations over time. Wright developed adaptive landscapes, populations climbing fitness peaks in multidimensional genetic combination landscape. Haldane calculated advantageous gene spread speed, showed even small selective advantages have large effects over evolutionary time. Abstract. Mathematical, difficult work but rigorous and powerful. We met Theodosius Dobzhansky, Russian-American geneticist, one of most important modern synthesis architects integrating genetics, natural selection, paleontology, systematics, botany into unified framework, trained in Russia studying ladybird beetles, moved to United States 1927 working with fruit fly geneticist Thomas Hunt Morgan, combined Morgan's genetic techniques with Fisher's theoretical insights and his own natural population understanding. 1937 published Genetics and the Origin of Species, translated population genetics mathematical theory into biologist-understandable language with concrete examples from wild fruit fly population research. We learned Dobzhansky showed natural populations harbor enormous genetic variation, far more than suspected, raw material for evolution, demonstrated seemingly trivial genetic differences could measurably affect survival and reproduction, studied population adaptation to local conditions, how geographic barriers lead to genetic divergence, how new species form through genetic incompatibility accumulation, made evolution tangible and testable, moving from abstract theory to observable process. We met Ernst Mayr, German ornithologist studying New Guinea and Solomon Islands bird species, published Systematics and the Origin of Species 1942, brought naturalists' eye to species formation question, emphasized species aren't just similar organism groups but reproductively isolated populations, don't interbreed with other species. Biological species concept clarified what species is and how new species arise. We found Mayr argued new species typically form when populations become geographically separated, mountain range rising, river changing course, few individuals colonizing island. In isolation separated populations evolve independently, accumulating different mutations, adapting to different local conditions, eventually become so different that even in contact can't successfully interbreed, become separate species. Allopatric speciation became dominant biodiversity model. We discovered paleontologist George Gaylord Simpson added deep time perspective. 1944 published Tempo and Mode in Evolution examining fossil record revelations about evolutionary patterns, showed fossils confirmed general picture from genetics and field biology, evolution occurred at varying rates, sometimes slowly/gradually, sometimes rapidly during environmental change or new habitat colonization. Fossil record gaps troubling Darwin explained by fossilization rarity and incomplete sampling. Not mysterious jumps or divine intervention. Each fact a gentle step in understanding's maturation. Darwin's theory tested and strengthened by mathematics, genetics, field observations, fossils, all coming together like streams flowing into a great river. The synthesis emerging gradually, patiently, as different minds contributed different pieces, different perspectives, building something larger than any individual could create alone. Rest in this pattern now, this beautiful convergence of knowledge. Let the synthesis wash over you like gentle waves, mathematics and biology, laboratory and field, all finding harmony, all flowing together toward deeper understanding. Let yourself drift with this flow, peaceful and calm, toward sleep. As we continue our peaceful journey through evolution's story, we arrive at the mid-20th century, when the modern synthesis, this beautiful integration of Darwin's natural selection with Mendelian genetics, became the foundation of biology. But scientific understanding never stands still. Even as this synthesis solidified, new questions emerged, new evidence appeared, and new debates began that would deepen and sometimes challenge the consensus that had been so carefully built. One of the most important contributions to the modern synthesis came from botany, from a scientist who studied plants with the same rigor that Dobzhansky and Mayr had brought to animals. G. Ledyard Stebbins was an American botanist who specialized in understanding variation and evolution in flowering plants. In 1950, he published Variation and Evolution in Plants, a comprehensive work that showed how the principles of the modern synthesis applied to the plant kingdom, which had often been neglected in favor of animals. Stebbins revealed that plants had their own unique evolutionary mechanisms that animals didn't share. Plants, unlike animals, could undergo polyploidy, a doubling or multiplication of their entire chromosome set, and this could create new species in a single generation. A plant with twice the normal number of chromosomes might be instantly reproductively isolated from its parent population, unable to produce fertile offspring with normal plants. This was evolution happening not gradually over many generations, but suddenly, in a single reproductive event. Stebbins showed that polyploidy had been crucial in plant evolution and that many crop plants humans depend on, wheat, cotton, strawberries, were polyploids, carrying multiple copies of ancestral genomes. With Stebbins's work, the modern synthesis was complete. It encompassed animals and plants, genetics and natural history, laboratory and field, mathematics and observation. By the 1950s, there was broad consensus among biologists. Evolution occurred through natural selection acting on genetic variation. Populations gradually adapted to their environments. Geographic isolation led to new species formation, and all life on Earth shared common ancestry stretching back billions of years. This framework would guide biology for decades to come. But even as this consensus formed, a new revolution was brewing in a different part of biology, one that would eventually transform evolution from an abstract theory about genes and populations into something concrete and molecular. In 1953, James Watson and Francis Crick, working at Cambridge University with crucial X-ray crystallography data from Rosalind Franklin, determined the structure of DNA, the famous double helix. This elegant molecule, with its two intertwined strands and complementary base pairing, immediately suggested how genetic information could be copied and passed from generation to generation. The discovery of DNA's structure opened a new era in biology. For the first time, scientists could see the physical basis of heredity, the actual molecule that carried genetic information. The abstract genes that Mendel had inferred from his pea-plant ratios, that Morgan had mapped on fruit fly chromosomes, that Fisher had modeled mathematically, were revealed to be sequences of chemical building blocks, adenine, thymine, guanine, and cytosine, arranged along the DNA molecule. Changes in these sequences, mutations, were the raw material of evolution, now visible at the molecular level. Throughout the 1960s, molecular biology exploded with discoveries. Scientists learned how DNA is transcribed into RNA and translated into proteins, the workhorses of the cell. They deciphered the genetic code, the rules by which triplets of DNA bases specify particular amino acids and proteins. They discovered that all living things, from bacteria to humans, use essentially the same genetic code, powerful evidence for the common ancestry of all life. The molecular mechanisms of life turned out to be far more complex than anyone had imagined, yet also far more unified, the same basic machinery in every living cell. For evolutionary biology, molecular data provided an entirely new kind of evidence. By comparing DNA or protein sequences between different species, scientists could measure evolutionary relationships with unprecedented precision. Species that diverged recently would have very similar sequences, differing only in a few positions. Species that diverged long ago would have accumulated many more differences. In 1965, Emil Zuckerkandl and Linus Pauling proposed the concept of the molecular clock, the idea that mutations accumulate at roughly constant rates over time, allowing DNA sequences to serve as a kind of evolutionary stopwatch. The molecular clock was revolutionary because it allowed scientists to estimate when different lineages diverged, even without fossils. By counting the differences in DNA or protein sequences between two species and assuming a constant mutation rate, researchers could calculate approximately when those species shared a common ancestor. This molecular dating complemented the fossil record, sometimes confirming paleontological estimates, sometimes surprising scientists with dates much older or younger than expected. The molecular clock wasn't perfect. Mutation rates varied somewhat between different genes and different lineages, but it provided a powerful new tool for reconstructing the tree of life. As molecular data accumulated through the 1960s and 1970s, a surprising picture emerged. At the molecular level, most genetic variations seemed to be neutral, neither beneficial nor harmful. In 1968, the Japanese geneticist Matou Kimura proposed the neutral theory of molecular evolution, arguing that the vast majority of mutations that become fixed in populations don't improve fitness but simply drift randomly to fixation because they don't affect survival or reproduction enough for selection to act on them. Kimura's neutral theory sparked intense debate. It seemed to challenge the centrality of natural selection, suggesting that much of evolution at the molecular level was driven by random genetic drift rather than adaptation. But this was actually a subtle point that many misunderstood. Kimura wasn't saying that natural selection was unimportant. He acknowledged that selection shaped organisms' adaptations, their visible traits, their interactions with the environment. Rather, he was pointing out that at the level of DNA sequences, many mutations were functionally equivalent, changing a DNA base here or there without affecting the protein's function, and these neutral changes accumulated through random chance. The neutral theory turned out to be enormously important for molecular evolution. It provided a null hypothesis, a baseline expectation for how DNA sequences should change over time if only random drift were operating. Deviations from this neutral expectation could reveal where natural selection had actually been at work, either preserving important sequences unchanged or driving rapid adaptive changes. The debate between neutralists and selectionists pushed evolutionary biology to become more rigorous, more quantitative, more careful about distinguishing different levels of biological organization. While molecular biology was revolutionizing laboratory science, field biologists were making their own crucial discoveries about how evolution actually happens in nature. One of the most influential field studies began in 1973, when Peter and Rosemary Grant, a husband-and-wife team of evolutionary biologists, began studying finches on a tiny island called Daphne Major in the Galapagos, the same islands Darwin had visited nearly 140 years earlier. The Grants' work was distinguished by its extraordinary patience and thoroughness. Year after year, decade after decade, they returned to this remote island, catching, measuring, and marking every single finch, thousands of individual birds. They measured beak size and shape with calipers, noted which birds survived and which died, tracked who mated with whom, recorded the seeds each bird ate. This was evolution in real time. Natural selection observed directly rather than inferred from fossils or genetic patterns. What the Grants discovered was remarkable. Natural selection was constantly at work, and it was far more dynamic than many had assumed. During drought years, when small soft seeds became scarce and only large hard seeds remained, birds with larger, Stronger beaks survived better. They could crack the tough seeds that smaller beaked birds couldn't handle. The average beak size in the population increased within a single generation. Then, when rains returned and small seeds became abundant again, selection reversed. Now smaller beaks were advantageous, and average beak size decreased. Evolution wasn't a slow, steady march in one direction but a constant fluctuation, populations tracking environmental changes that occurred over months and years, not just millennia. The grants also documented the formation of hybrids, finches of different species occasionally interbreeding, and showed that these hybrids could sometimes survive and reproduce, introducing genetic variation from one species into another. This gene flow between species added complexity to the picture of evolution, showing that the boundaries between species could be more porous than the classical species concept suggested. The grant's work, published over decades in meticulous detail, provided some of the most compelling direct evidence for evolution by natural selection ever gathered, Darwin's theory verified in Darwin's own finches. And so we met G. Ledyard Stebbins, American botanist who in 1950 published Variation and Evolution in Plants, comprehensive work showing modern synthesis principles applied to plant kingdom, often neglected for animals, revealed plants had unique evolutionary mechanisms animals didn't share, particularly polyploidy, doubling or multiplying entire chromosome set creating new species in single generation. Plant with twice normal chromosomes instantly reproductively isolated from parent population. Unable to produce fertile offspring with normal plants, evolution happening suddenly in single reproductive event, not gradually over generations. We learned Stebbins showed polyploidy crucial in plant evolution. Many crop plants humans depend on, wheat, cotton, strawberries, were polyploids carrying multiple ancestral genome copies. With Stebbins's work modern synthesis was complete, encompassing animals and plants, genetics and natural history, laboratory and field, mathematics and observation. By 1950s broad consensus among biologists, evolution occurred through natural selection acting on genetic variation, populations gradually adapted, geographic isolation led to new species, all life shared common ancestry stretching billions of years. We witnessed 1953's new revolution, James Watson and Francis Crick at Cambridge University, with crucial X-ray crystallography data from Rosalind Franklin, determined DNA structure, famous double helix, elegant molecule with two intertwined strands and complementary base pairing immediately suggested how genetic information copied and passed generation to generation, open new era. First time scientists could see heredity's physical basis, actual molecule carrying genetic information. We discovered abstract genes Mendel inferred from P ratios, Morgan mapped on fruit fly chromosomes, Fisher modeled mathematically, revealed as sequences of chemical building blocks, adenine, thymine, guanine, cytosine, arranged along DNA molecule. Changes in these sequences, mutations, were evolution's raw material, now visible at molecular level. We followed 1960s molecular biology explosion. Scientists learned how DNA transcribed into RNA, translated into proteins, cells' workhorses, deciphered genetic code, rules by which DNA-based triplets specify particular protein amino acids, discovered all living things, bacteria to humans, use essentially same genetic code, powerful common ancestry evidence, molecular life mechanisms far more complex than imagined yet far more unified, same basic machinery in every living cell. We learned molecular data provided entirely new evolutionary evidence. Comparing DNA or protein sequences between species measured evolutionary relationships with unprecedented precision. Recently diverged species, very similar sequences, few differences, long ago diverged, many differences. 1965 Emil Zuckerkandl and Linus Pauling proposed molecular clock. Mutations accumulate at roughly constant rates over time, allowing DNA sequences to serve as evolutionary stopwatch. We found molecular clock revolutionary because allowed estimating lineage divergence times without fossils, counting DNA/protein sequence differences between species and assuming constant mutation rate calculated approximately when they shared common ancestor. Molecular dating complemented fossil record, sometimes confirming paleontological estimates, sometimes surprising with much older/younger dates. Not perfect, mutation rates varied between genes and lineages, but powerful new tool for reconstructing life's tree. We discovered 1960s to 1970s molecular data revealed surprising picture. At molecular level, most genetic variations seemed neutral, neither beneficial nor harmful. 1968 Japanese geneticist Matu Kimura proposed neutral theory of molecular evolution. Vast majority of mutations becoming fixed in populations don't improve fitness but drift randomly to fixation because don't affect survival/reproduction enough for selection to act. Sparked intense debate seeming to challenge natural selection's centrality, suggesting much molecular evolution driven by random genetic drift rather than adaptation. We learned Kimura's subtle point many misunderstood. Wasn't saying natural selection unimportant, acknowledged selection-shaped adaptations, visible traits, environmental interactions. Rather, at DNA sequence level, many mutations functionally equivalent, changing DNA bases without affecting protein function. These neutral changes accumulating through random chance. Neutral theory provided null hypothesis, baseline expectation for DNA sequence changes under only random drift, deviations revealed. Where natural selection actually worked, preserving important sequences or driving rapid adaptive changes. We met Peter and Rosemary Grant, husband-wife evolutionary biologist team beginning 1973 studying finches on tiny Galapagos island Daphne Major, same islands Darwin visited nearly 140 years earlier, work distinguished by extraordinary patience and thoroughness, year after year, decade after decade returning, catching, measuring, marking every single finch, thousands of individuals, measured beak size/shape with calipers, noted survivors/deaths, tracked mating, recorded seeds eaten, evolution in real time, natural selection observed directly not inferred. We discovered Grant's remarkable findings. Natural selection constantly at work, far more dynamic than assumed. Drought years when small soft seeds scarce, only large hard seeds remained. Birds with larger stronger beaks survived better, could crack tough seeds. Average population beak size increased within single generation. When rains returned and small seeds abundant, selection reversed, smaller beaks advantageous. Average size decreased, evolution not slow steady march but constant. Fluctuation, populations tracking environmental changes over months and years, not just millennia. We learned Grant's documented hybrid formation, different species finches occasionally interbreeding, hybrids sometimes survived and reproduced, introducing genetic variation from one species into another. Gene flow between species added complexity, showed species boundaries more porous than classical concepts suggested. Grant's work over decades provided most compelling direct evidence for evolution by natural selection ever gathered, Darwin's theory verified in Darwin's own finches. Each fact a gentle reminder of how understanding deepens, Darwin's insights confirmed at molecular level, natural selection observed in real time, neutral changes and adaptive changes both part of evolution's rich tapestry. The synthesis growing stronger through testing, through observation, through patient accumulation of evidence across scales from molecules to ecosystems. Rest now in this deepening understanding, this recognition that truth reveals itself in layers, molecules and organisms, laboratories and islands, mathematics and measurement all telling the same story in different voices. Let yourself drift with these discoveries, peaceful and calm, carried gently toward sleep. As we continue our peaceful journey through evolution's story, we move into the final decades of the 20th century and the opening of the 21st, a time when evolutionary biology would expand in directions Darwin could never have imagined, yet always returning to the fundamental insights he first articulated aboard the Beagle and in his quiet study at Down House. One of the most significant developments came in 1972, when two young paleontologists, Niles Eldredge and Stephen Jay Gould, proposed a theory they called punctuated equilibrium. They had been studying fossils and noticed a puzzling pattern that had troubled paleontologists for decades. Most species in the fossil record appear suddenly, persist unchanged for millions of years, then disappear, rather than showing the gradual transformation Darwin's theory seemed to predict. Darwin himself had attributed this pattern to the incompleteness of the fossil record. The rarity of fossilization and the accidents of preservation meant we were seeing only snapshots, missing the gradual transitions. But Eldredge and Gould suggested something different. Perhaps the pattern was real, not an artifact of poor preservation. They proposed that most evolutionary change occurs rapidly during speciation events, when new species form, and that once established, species remain relatively stable for long periods. This didn't contradict Darwin's theory of gradual change, but it relocated where and when that change occurred. Instead of slow, steady transformation throughout a species' existence, most change happened in small isolated populations during the splitting of lineages, and these events were geologically rapid, taking perhaps thousands of years rather than millions, but still gradual when measured in generations. Punctuated equilibrium sparked fierce debate. Some saw it as a revolutionary challenge to Darwinian orthodoxy. Others saw it as a minor modification, a matter of tempo and mode rather than mechanism. Gould himself became a public intellectual, writing eloquent essays about evolution for general audiences, always emphasizing the role of contingency and chance in life's history. The debate pushed paleontologists and population geneticists to talk to each other more, to think more carefully about how processes operating over hundreds of generations might appear in fossils recording events millions of years apart. While debates about evolutionary tempo continued, another revolution was quietly transforming biology: the ability to read and manipulate DNA sequences directly. By the 1980s, techniques for sequencing DNA, determining the exact order of A's, T's, G's, and C's along the molecule, were becoming faster and more affordable. Scientists could now compare genes from different species with precision that would have seemed like science fiction just decades earlier. These comparisons revealed evolution's signature written in every organism's genome. The more DNA sequences scientists examined, the more clearly they could see the tree of life. All mammals shared certain genetic sequences that differed from those in birds. All vertebrates shared sequences that differed from those in insects. All animals shared sequences that differed from those in plants. And underlying it all life, from bacteria to redwood trees to human beings, shared fundamental genetic machinery, the same basic genes for reading DNA and making proteins, the same genetic code, variations on the same molecular themes. This was exactly what Darwin's theory predicted: all life sharing common ancestry, the degree of genetic similarity reflecting how recently lineages had diverged. In 1990, an international collaboration launched the Human Genome Project, an ambitious effort to sequence all 3 billion base pairs of human DNA. At the time, many thought the project was impossibly ambitious, perhaps a waste of resources. But new technologies accelerated the pace of sequencing, and by 2003, the human genome was essentially complete. For the first time, humanity could read its own genetic instruction book, see the genes that make us human written in DNA's 4-letter alphabet. The human genome revealed humbling truths. Humans have only about 20,000 protein-coding genes, not so many more than a fruit fly or a roundworm. Much of our DNA doesn't code for proteins at all, and some of it appears to be remnants of ancient viral infections or copies of genes that have lost their function, molecular fossils of our evolutionary past. Our genome carries the signatures of our history, broken vitamin C synthesis genes we share with other primates who get vitamin C from fruit, deactivated genes for producing egg yolk proteins we share with egg-laying ancestors, genetic sequences remarkably similar to chimpanzees, our closest living relatives. The comparison between human and chimpanzee genomes was particularly revealing. In 2005, the chimpanzee genome was sequenced, and scientists could finally quantify how similar we are to our closest relatives. The answer: about 98 to 99% identical in the DNA sequences we share. This tiny genetic difference, perhaps 1 to 2% of our genomes, accounts for all the visible differences between humans and chimps: our larger brains, our upright posture, our capacity for language, our reduced body hair. Such a small genetic change producing such profound differences showed how changes in gene regulation, when and where genes are turned on and off, can be as important as changes in the genes themselves. These genomic comparisons also allowed scientists to estimate when human and chimpanzee lineages diverged from their common ancestor. The molecular clock, calibrated with fossil evidence, suggested the split occurred about 6 to 7 million years ago. This meant that somewhere in Africa, around that time, a population of apes divided, one lineage eventually leading to modern chimpanzees and bonobos, the other to the various species of hominins, including eventually Homo sapiens. The human evolutionary story could now be told not just through fossils but through genes, two independent lines of evidence converging on the same narrative. As genomic data accumulated through the late 1990s and 2000s, scientists made discoveries that would have seemed impossible just years earlier. One of the most remarkable was the recovery and sequencing of DNA from extinct species. For decades, the conventional wisdom was that DNA degraded too quickly after death to be recovered from ancient fossils. But in 2010, an international team led by Svana Pabo announced they had sequenced the genome of Neanderthals, human relatives who went extinct about 40,000 years ago, using DNA extracted from ancient bones. The Neanderthal genome revealed something unexpected. Modern humans of European and Asian ancestry carry 1 to 2% Neanderthal DNA, while sub-Saharan Africans carry essentially none. This could mean only one thing. After early humans left Africa and spread into Europe and Asia, they encountered Neanderthals and interbred with them. Those ancient liaisons, tens of thousands of years ago, left traces in our genomes that we carry today. Evolution wasn't a simple branching tree with separate species on isolated branches. Sometimes the branches touched, and genes flowed between species, adding complexity and richness to the story of our origins. The recovery of ancient DNA opened an entirely new field called paleogenomics, the study of genetic material from extinct organisms. Scientists sequenced the genomes of woolly mammoths, cave bears, giant ground sloths, and even enigmatic hominins known only from a few bone fragments in a Siberian cave called Denisova. These Denisovans, as they came to be called, were identified primarily from their DNA rather than their fossils, a new species discovered in the genetic code rather than through bones and teeth. Modern humans of Asian and Oceanian ancestry carry Denisovan DNA, evidence of yet another ancient interbreeding event. These discoveries transformed our understanding of human evolution from a simple linear progression to a complex braided stream, with multiple hominin species coexisting, sometimes interbreeding, their genetic legacies mixing and flowing through time. Evolution was messy, more intricate than earlier models suggested, but also more wonderful, a story not of isolated species marching separately through time, but of populations connecting, diverging, sometimes reconnecting, weaving together the rich tapestry of life's diversity. By the early 2000s, the cost of DNA sequencing was plummeting. What had cost billions of dollars for the first human genome could be done for thousands, then hundreds. This democratization of genomics allowed evolutionary biologists to study evolution in unprecedented detail across thousands of species. Scientists could trace how genes evolved, which genes were conserved across vast evolutionary distances, which genes showed signs of recent rapid evolution, how genes were gained, lost, duplicated, and repurposed throughout life's history. One particularly beautiful application was the study of evolutionary developmental biology, EVO-DEVO for short, which examined how changes in development produce evolutionary novelty. Researchers discovered that the same toolkit of developmental control genes, like the famous HOX genes that specify body segment identity, was shared across enormously diverse animals. A gene that helps pattern a fly's body segments has a similar counterpart helping pattern a mouse's spine. These deep genetic similarities revealed that evolution often works by tinkering with ancient genetic programs, changing when and where they're activated, rather than inventing entirely new genes. The EVO-DEVO perspective showed how small genetic changes early in development could produce large morphological changes in the adult organism. A mutation affecting where limb development genes are expressed might move a fin slightly forward on a fish's body, or change the number of digits on a developing hand. This helped explain one of evolution's enduring mysteries: how major transitions occur, how fish evolved limbs and moved onto land, how reptiles evolved feathers and took to the air, how whales evolved from land mammals and returned to the sea. These weren't miraculous leaps but accumulations of small changes in developmental programs, each step functional, each step selected. And so we met Niles Eldredge and Stephen Jay Gould, who in 1972 proposed punctuated equilibrium. Studying fossils noticed puzzling pattern troubling paleontologists for decades. Most species appear suddenly, persist unchanged for millions of years, then disappear, rather than showing gradual transformation Darwin's theory seemed to predict. Darwin attributed this to incomplete fossil record, fossilization rarity, preservation accidents, but Eldredge and Gould suggested pattern was real, not preservation artifact. We learned punctuated equilibrium proposed most evolutionary change occurs rapidly during speciation events. New species formation, once established, species remain relatively stable for long periods, didn't contradict gradual change but relocated where and when it occurred, instead of slow steady transformation throughout species existence. Most change happened in small isolated populations during lineage splitting, geologically rapid, thousands of years not millions, but still gradual in generations. We found punctuated equilibrium sparked fierce debate. Some saw revolutionary challenge to Darwinian orthodoxy, others saw minor modification of tempo and mode not mechanism. Gould became public intellectual, writing eloquent evolution essays emphasizing contingency and chance in life's history. Debate pushed paleontologists and population geneticists toward more communication, careful thinking about how processes over hundreds of generations might appear in fossils recording millions of years apart events. We discovered 1980s revolution, ability to read and manipulate DNA sequences directly, DNA sequencing techniques, determining exact A, T, G, C order, becoming faster and more affordable. Scientists could compare genes from different species with unprecedented precision. Comparisons revealed evolution's signature written in every organism's genome. All mammals shared certain sequences differing from birds. All vertebrates shared sequences differing from insects. All animals shared sequences differing from plants. All life shared fundamental genetic machinery. We followed 1990 international collaboration launching Human Genome Project, ambitious effort sequencing all 3 billion human DNA base pairs. Many thought impossibly ambitious, resource waste, but new technologies accelerated sequencing. By 2003, human genome essentially complete. First time humanity could read its own genetic instruction book. Genes making us human written in DNA's 4-letter alphabet. We learned human genome revealed humbling truths. Humans have only 20,000 protein coding genes, not many more than fruit fly or roundworm. Much DNA doesn't code for proteins. Some appears to be ancient viral infection remnants or lost function gene copies, molecular fossils of evolutionary past. Genome carries history signatures. Broken vitamin C synthesis genes shared with fruit-eating primates. Deactivated egg yolk protein genes from egg-laying ancestors. Sequences remarkably similar to chimpanzees. We discovered 2005 chimpanzee genome sequence allowed quantifying our similarity to closest relatives, 98-99% identical in shared DNA sequences. Tiny 1 to 2% genetic difference accounts for all visible human-chimp differences, larger brains, upright posture, language capacity, reduced body hair. Such small genetic change producing profound differences showed gene regulation changes, where genes turn on off, can be as important as gene changes themselves. We found genomic comparisons allowed estimating human-chimpanzee lineage divergence from common ancestor. Molecular clock calibrated with fossils suggested split 6-7 million years ago. Somewhere in Africa around that time, ape population divided, one lineage eventually leading to modern chimps and bonobos, other to various hominin species including Homo sapiens. Human evolutionary story now told through fossils and genes, two independent evidence lines converging on same narrative. We witnessed late 1990s to 2000s genomic data accumulation making impossible seeming discoveries. 2010 international team led by Svante Pääbo announced sequencing Neanderthal genome, human relatives extinct 40,000 years ago, using ancient bone DNA. Conventional wisdom said DNA degraded too quickly after death for ancient fossil recovery. But Neanderthal genome revealed unexpected finding. Modern humans of European/Asian ancestry carry 1 to 2% Neanderthal DNA, sub-Saharan Africans essentially none. We learned this meant after early humans left Africa spreading into Europe/Asia, they encountered Neanderthals and interbred. Ancient liaisons tens of thousands of years ago left traces in genomes we carry today. Evolution wasn't simple branching tree with separate species on isolated branches, sometimes branches touched, genes flowed between species, adding complexity and richness to origin story. We discovered ancient DNA recovery opened new paleogenomics field, genetic material study from extinct organisms, scientists sequenced woolly mammoths, cave bears, giant ground sloths, enigmatic Siberian cave, Denisova, hominins known from few bone fragments, Denisovans identified primarily from DNA rather than fossils, new species discovered in genetic code not through bones/teeth. Modern humans of Asian/Oceanian ancestry carry Denisovan DNA, evidence of another ancient interbreeding event. We found discoveries transformed human evolution understanding from simple linear progression to complex braided stream, multiple hominin species coexisting, sometimes interbreeding, genetic legacies mixing and flowing through time, evolution Messier, more intricate than earlier models but more wonderful, story not of isolated species marching separately but populations connecting, diverging, sometimes reconnecting, weaving life's diversity tapestry. We witnessed early 2000s DNA sequencing cost plummeting, what cost billions for first human genome done for thousands then hundreds. Genomics democratization allowed studying evolution in unprecedented detail across thousands of species, traced how genes evolved, which conserved across vast distances, which showed recent rapid evolution, how genes gained, lost, duplicated, repurposed throughout life's history. We explored evolutionary developmental biology, evo-devo, examining how developmental changes produce evolutionary novelty. Researchers discovered same developmental control gene toolkit, like HOX genes specifying body segment identity, shared across enormously diverse animals. Gene helping pattern fly body segments has similar counterpart helping pattern mouse spine. Deep genetic similarities revealed evolution often works by tinkering with ancient genetic programs, changing when/where activated, not inventing entirely new genes. We learned evo-devo perspective showed small early developmental genetic changes could produce large adult morphological changes. Mutation affecting limb development gene expression location might move fish fin forward or change developing hand digit number, helped explain major transitions, fish evolving limbs moving onto land, reptiles evolving feathers taking to air, whales evolving from land mammals returning to sea. Not miraculous leaps but small developmental program change accumulations. Each step functional and selected. Each fact a gentle reminder of how understanding grows richer and more complex. Evolution revealed not just in fossils but in DNA, not just between species but within our own genomes, not just as history but as ongoing process written in molecular code. The story deepening with each discovery, each technological advance, each new way of looking at life's grand pattern. Rest now in this rich complexity, this beautiful intricacy of life's history written in molecules and bones, in genes and fossils, in ancient DNA and living populations. Let the story carry you, support you, cradle you gently towards sleep, where your own ancient genetic heritage dreams its way forward into tomorrow. As we approach the end of our peaceful journey through evolution's story, we arrive at the present moment, a time when evolutionary biology has become more vibrant, more interconnected, and more relevant than ever before. The science that began with Darwin's patient observations of finches and barnacles now encompasses everything from medicine to conservation, from agriculture to understanding our own human nature. One of the most exciting frontiers in modern evolutionary biology is the study of evolution happening right now, in real time, often driven by human activities. Scientists have documented countless examples of rapid evolution in response to human-caused environmental changes. One of the most striking involves the peppered moth in industrial England, a classic example taught in biology classes, but one whose details are worth savoring in their elegant simplicity. Before the Industrial Revolution, peppered moths in England were mostly light-colored with dark speckles, camouflaged against lichen-covered trees. But as factories proliferated and coal smoke darkened the tree trunks, killing the lichens, light-colored moths became conspicuous against the dark bark. Birds could easily spot and eat them. Dark-colored moths, which had always existed in small numbers due to rare mutations, now had the advantage. They were camouflaged against the soot-covered trees. Within just 50 years, from roughly 1800 to 1850, dark moths became the common form in industrial areas, comprising over 90% of the population in some regions. This shift was evolution by natural selection, observed and documented as it happened. Then, in the late 20th century, as pollution controls cleaned the air and lichens returned to the trees, the process reversed. Light-colored moths once again had the advantage, and their frequency increased. The peppered moth became a beautiful demonstration of evolution's responsiveness, populations tracking environmental changes over mere decades, natural selection visible within a human lifetime. But evolution in response to human activities isn't limited to moths. Antibiotic resistance in bacteria is evolution happening at terrifying speed, with profound implications for human health. When antibiotics were first introduced in the 1940s, penicillin and streptomycin, they seemed like miracle drugs, capable of curing infections that had killed millions throughout human history. But bacteria reproduce so rapidly, and their populations are so large, that rare mutations conferring resistance arise frequently. When antibiotics are used, they create powerful selection pressure. Bacteria without resistance die, but the rare resistant individuals survive and reproduce. Within years of penicillin's introduction, resistant bacteria appeared. The same happened with every new antibiotic, initial effectiveness followed by the gradual or sometimes rapid emergence of resistance. Today, antibiotic resistance is a global health crisis. Bacteria have evolved resistance to multiple antibiotics, creating superbugs that are extremely difficult to treat. This is Darwinian evolution in action, a stark reminder that we share our planet with organisms that evolve far faster than we do, organisms that will always adapt to whatever challenges we throw at them. Understanding evolution has become essential for medicine in other ways too. Cancer, it turns out, is an evolutionary process happening within our own bodies. Cancer begins when a cell acquires mutations that allow it to escape normal growth controls. That cell divides, and its descendants divide, creating a population of cancer cells. Within this population, further mutations arise, and the cancer cells that grow fastest evade the immune system most effectively, or resist chemotherapy drugs will be selected for. A tumor is an evolving population, shaped by natural selection. This evolutionary perspective on cancer has transformed how oncologists think about treatment. If you treat a tumor with chemotherapy, you're applying selection pressure. You'll kill most of the cancer cells, but any cells with mutations conferring drug resistance will survive and proliferate. The cancer will evolve resistance, just as bacteria evolve antibiotic resistance. Understanding this has led to new treatment strategies, using multiple drugs simultaneously to make resistance less likely, varying treatment protocols to change selection pressures, even deliberately allowing some cancer cells to survive to compete with more aggressive ones. Cancer treatment has become an evolutionary arms race, and evolutionary biologists now work alongside medical researchers to develop more effective therapies. The evolutionary perspective has also revolutionized our understanding of aging and life history, why organisms age at different rates, why some reproduce once and die while others reproduce throughout long lives, why different species allocate resources differently between growth, reproduction, and longevity. These questions, which seemed purely descriptive, turn out to have deep evolutionary explanations rooted in trade-offs and selection pressures across different life stages. Life history theory, developed primarily in the 1960s through 1980s, explains these patterns through the lens of natural selection optimizing reproductive success. If an organism lives in a dangerous environment where death from predation or disease is likely, selection favors early reproduction, better to reproduce young and pass on genes before death comes. This leads to rapid maturation, early reproduction, often many offspring, and shorter lifespan. But in safer environments, where survival to old age is more likely, selection can favor delayed reproduction, fewer but better cared for offspring, and longer lifespans that allow multiple reproductive events. These principles explain why mice reproduce prolifically and live 2 years while elephants reproduce slowly and live 70, not because elephants are better but because their different ecological situations favor different life history strategies. The same principles apply to plants. Annual plants that live in disturbed habitats germinate, grow, reproduce, and die in a single season, while trees in stable forests grow slowly, reproduce over many years, and live for centuries. Each strategy is optimal for its particular environment, shaped by millions of years of natural selection. Understanding life history evolution has practical applications for conservation biology. When we try to save endangered species, we need to understand their life history strategies. A species that reproduces slowly with few offspring, like giant pandas or great whales, cannot quickly recover from population declines. They require long-term protection and careful management. Species that reproduce prolifically can bounce back more quickly but may be more vulnerable to environmental instability. Conservation strategies must be tailored to each species' evolved life history, taking into account millions of years of evolution that shaped how they live and reproduce. Another frontier where evolutionary biology has become crucial is understanding emerging infectious diseases. The COVID-19 pandemic that began in 2019 was a stark reminder that evolution doesn't stop, that new diseases can emerge when pathogens jump from animal hosts to humans, and that viruses evolve with frightening speed. The SARS-CoV-2 virus that causes COVID-19 likely originated in bats, jumped to humans possibly through an intermediate host, and then rapidly evolved as it spread through human populations. Tracking the virus's evolution became central to managing the pandemic. Scientists sequenced viral genomes from infected patients around the world, watching in real time as mutations arose and spread. Some mutations were neutral, accumulating through random drift. But some changed the virus's properties, making it more transmissible, helping it evade antibodies, potentially affecting disease severity. New variants emerged, Alpha, Delta, Omicron, each representing a lineage that had evolved advantageous mutations and outcompete previous variants. Understanding viral evolution-guided vaccine development helped predict which variants might become dominant and informed public health responses. This wasn't the first time evolutionary biology proved essential for understanding disease. The principles apply to influenza, which evolves so rapidly that new vaccines must be developed each year to match circulating strains. They apply to HIV, which evolves within each infected patient, quickly developing resistance to antiviral drugs. They apply to malaria parasites, tuberculosis bacteria, and countless other pathogens. Evolutionary biology isn't an abstract academic subject, it's a vital tool for protecting human health, for understanding the living world we're inextricably part of. Perhaps most profoundly, evolutionary biology has deepened our understanding of ourselves, our bodies, our behavior, our place in nature. We are not separate from the natural world but products of it, shaped by the same evolutionary forces that shaped every other living thing. Our large brains, our capacity for language and abstract thought, our complex social behaviors, our emotions and instincts, all are products of evolution, adaptations that helped our ancestors survive and reproduce in the environments they inhabited. Evolutionary psychology attempts to understand human behavior through this lens, asking which psychological traits might be adaptations and how they might have been selected. Why do we find certain landscapes beautiful, particularly savanna-like environments with scattered trees, water sources, and distant views? Perhaps because these were the environments where our ancestors evolved, and individuals attracted to such landscapes were more likely to find resources and survive. Why do we form close bonds with family and reciprocal relationships with non-relatives? Because cooperation and kinship helped our ancestors survive in social groups. These are tentative explanations, hypotheses to be tested, not just stories to be accepted uncritically. But they represent a profound shift in how we think about ourselves, not as beings separate from nature, designed by divine intention, but as products of natural processes, evolved animals whose every trait, from our upright posture to our sense of humor, has a history stretching back millions of years. And so our journey through evolution's story comes to its end, though the story itself continues, unfolding moment by moment in every living thing on Earth. We've traveled from Erasmus Darwin's speculative musings to modern genomics, from young Charles Darwin aboard the Beagle to the architects of the modern synthesis, from the discovery of DNA's structure to the sequencing of Neanderthal genomes, from debates in Victorian Oxford to real-time tracking of viral evolution. We've seen how understanding grows, slowly at first, through patient observation and careful thought, then accelerating as new tools and technologies open new windows into life's history. We've watched as separate strands of evidence, fossils and genes, anatomy and molecules, laboratory experiments and field observations, all converge on the same magnificent story. Life on Earth shares common ancestry, has changed over billions of years, has diversified into millions of forms through natural selection acting on random variation. This story is not complete. There are still mysteries to solve, debates to settle, discoveries waiting to be made. We don't fully understand how the first living cells arose from non-living chemistry. We're still mapping the tree of life, still discovering new species, still finding surprises in genomes and fossils. We're still learning how development translates genes into organisms, how ecosystems evolve, how consciousness emerged from neural tissue. The story of evolution is ongoing, and future generations will add chapters we can't yet imagine. But the fundamental framework is secure, tested and refined over 160 years since Darwin published The Origin of Species. Evolution is both a fact and a theory, a fact in that life has changed over time, that species share common ancestors, that the diversity of life arose through natural processes. And a theory in the scientific sense, a comprehensive explanatory framework, supported by overwhelming evidence from every branch of biology, that explains how evolution occurs through natural selection, genetic drift, and other mechanisms acting on inherited variation. This understanding connects us to all life on Earth in the most literal way. The bacteria in your gut, the trees outside your window, the birds you hear singing, they are your relatives, your cousins, sharing with you a common ancestor that lived billions of years ago. The same DNA alphabet writes instructions in every living cell. The same basic biochemistry animates bacteria and butterflies, mushrooms and humans. We are all part of one grand living tapestry, woven by 4 billion years of evolution, still being woven in this present moment. As you rest now, drifting towards sleep, you carry within you this 4 billion year legacy. Your cells contain genes inherited from fish ancestors, from reptile ancestors, from small furry mammals hiding from dinosaurs, from primates in ancient forests, from the first humans in Africa. You are a brief flowering of this ancient lineage, consciousness contemplating its own origins, life becoming aware of itself and its history. Let that awareness be gentle, peaceful, without burden. You need understand nothing more tonight. The questions can wait. The mysteries will keep. For now, there is only rest, only the quiet recognition that you are part of something vast and ancient and ongoing, part of life's great story, connected to everything that has ever lived and everything that will live after you. The universe gave rise to stars, stars forged heavy elements, elements formed planets, chemistry became biology, simple cells became complex, evolution created diversity, awareness emerged, and here you are, stardust that learned to think, atoms arranged in such a way that they can contemplate their own existence. That alone is miraculous, whether or not we fully understand it. Rest now in that miracle. Let evolution's gentle story carry you into sleep, into dreams, into the mystery of consciousness fading and returning. 4 billion years of life have led to this moment, to you, here, now, peaceful and whole. Good night. Sleep well. The story continues next video.